Okay, so chapter 12 is nervous tissue. So it goes over basically the cells of the nervous system, neuron physiology, what happens at synapses, and how neurons are integrated. So the general functions of the nervous system are to, one, collect information, and we do that with receptors. So receptors are specialized structures, and what they do is they're going to monitor stimuli or what's going on in the external environment. Okay, so receptors like your eyes or their pain receptors, taste. So there's all sorts of different kinds of receptors. And we talk about those when we start talking about the eye and the senses. Okay, so after we receive or collect the information, then we're going to process it and evaluate it. So that can happen in the brain, it can happen in the spinal cord. It depends on what information is coming in. Hold on, okay. Then we initiate a response to the information. So the effectors that actually carry out the response from the nervous system are all three types of muscles and glands and other neurons. Okay, so that's how we're going to respond. Okay, so the subdivisions, so the structural or the anatomical subdivisions of the nervous system, so they consist of the central nervous system, so which is the brain and the spinal cord, and they're shown in this peachy color over here. And then the peripheral nervous system, which is yellow. Okay, so the peripheral nervous system is everything outside of the brain and the spinal cord. So it consists of nerves and it consists of ganglia. So what are nerves and ganglia? So a nerve is a bundle of nerve fibers. So it's a bunch of different axons all bundled together. You can think of it as a big, like if you took spaghetti and you taped it up and then you put a whole bunch of spaghetti together. That's what a nerve is. And then a ganglion is the cell body of the neurons, the cell body. So we'll talk about that. I'll show you a picture. Okay, so now how is it organized functionally? So anatomists and physiologists kind of, they explain things with anatomy and they explain things with physiology. So and anatomically, here's the last picture. Functionally, it's how the information is being processed. Okay, so sensory information is afferent and it's coming towards the central nervous system. Motor information is efferent and it's exiting away from the central nervous system. So efferent is E, so E and E. Okay, so sensory is coming in and efferent is leaving. Okay, somatic is we're aware of it, so it's coming from your body. So somatic is the motor division, so it's how we actually perceive things. So we're conscious of it. And visceral things are things that are autonomic, so they're coming from deep in the viscera, so deep in the body. So those are coming from within the thoracic or the abdominal cavity and within the blood vessels. Okay, so here's functional classes of neurons. So this is supposed to show you how the loop or the circuit kind of works. Okay, so here's the sensory. So say you cut your finger or you put it on a piece of broken glass. So the sensory or the afferent neurons are going to take the information into the central nervous system. In the central nervous system, there are some neurons that are called interneurons. Okay, so those would be within the CNS. Okay, and then the motor information is going to come back out. So when it's in the CNS, that's when the decision is going to be made as to how you react. So some things are autonomic or a reflex and you're not going to think about it. So like when you touch a hot stove, okay, or the hot loop in micro class. Okay, so you touch it, okay, the information comes in, goes to the central nervous system. It's a reflex or autonomic, so it will come back right back out. And that you pull your finger away before you even realize that it's hot. So the motor neurons, this is the efferent aspect. Okay, so this is showing you functionally, it's more in a chart. So sensory is going to detect the stimulus and takes the information to the central nervous system. Okay, so somatic is coming from the body, and we're consciously perceiving it. So it's stuff that we see, stuff that we feel, stuff that we hear. Okay, so we understand that it's going on. Okay, visceral sensory is things that are coming from your heart, from your lungs, from your blood vessels. So you are not conscious of it happening. So it's not consciously perceived. So it's autonomic. It's going to happen without you thinking about it. Okay, now motor nervous system. Okay, the information is coming back out. Okay, so we have somatic. Okay, so somatic is if you decide to move something, it's going to the skeletal muscle. Okay, so it's voluntary. Skeletal muscle is always going to be voluntary. And then autonomic is stuff that we're not thinking about. Okay, so it's how our body responds automatically. Okay, so the things that are going to cause the change there are cardiac muscle, smooth muscle, and glands. So if you think about it, you're not ever really thinking about, is my heart beating, or I need to beat my heart now, or I need to digest my food, or I need to release insulin. Okay, those are all autonomic motor responses. I hope that makes sense. So it's divided into two, sensory and somatic and visceral, and then motor and somatic and autonomic. So sensory's going in, motor's coming out. If you look at a neuron, if you ever found a cell, okay, and you had to say, what kind of cell is this? How would you characterize it as being a neuron? So the way that you would do that is you're going to look for these big things. So the first one I have is excitability. Okay, so a neuron can respond to stimulation. Okay, so it's going to respond to something happening to it. It can initiate a response, and the stimulation totally depends on where the neuron is. Okay, so if it's in your eye, it'll be something visual. If it's on your skin, it could be touch. Okay, neurons can conduct an electrical impulse. Okay, so that's how neurons work. Without that electrical impulse, they don't function. Okay, they can secrete, and what they secrete is they secrete neurotransmitters, which we'll abbreviate NT. They last a really, really long time. So you form them when you're a fetus, and if nothing happens to them, they'll last the rest of your life, as long as they're not damaged. Okay, and they don't typically divide after fetal development. So you usually, that's the problem, right? So if a neuron is damaged, the sensory information is not going to come in, and the motor information isn't going to go back out. So we haven't been able, if someone could figure out how to make them undergo mitosis, it would solve a lot of problems. But it doesn't exist yet. Okay, so if we go over the structure of the neuron, I'm going to write things in here. Okay, so the soma is where we're going to start, and that's the cell body. Okay, so this is the body of the neuron. Okay, the parts that extend off of that, these are called dendrites. So it's kind of like dendrite. The study of trees is dendrology, so that's supposed to be a D. So dendrites are the branches coming off of it. Okay, so they're cytoplasmic extensions. They're going to receive the stimulus. Okay, so they're going to take the information that's coming to the neuron. Okay. The nucleus, all the normal organelles are going to be found in the cell body. Okay, at the end of the cell body, this is called the axon hillock. Okay, and it's like a little bump at the end, and that's the trigger zone. That's where we're going to sum up the information that we're receiving and decide whether we're going to carry out an impulse or not. Okay, this is the axon. Okay, so that's the long part. And then down on this end, these are synaptic knobs or bulbs. They call them either thing. Inside of the cell body, neurons have their own modified cytoskeleton, so that has neurofibrils. So that's their specific kind of cytoskeleton. They have lots of actin in it. And they also have these things called Nissl bodies. So Nissl bodies are aggregates of rough endoplasmic reticulum, so they can make neurotransmitters. Okay, so this is going to form a junction with the next one. So if here's more dendrites coming off, and here's the cell body, okay, this one is going to connect and talk to this one. So this is the synaptic cleft. Okay, this neuron has myelin on it. So the myelin is the white stuff. Okay, and it's wrapped around it. We'll talk about where it comes from. And I think that's pretty good for right now. Okay, I'll make it green, I guess. Okay, so the neurotransmitters, the proteins are going to be made here, but they have to travel down this way. Okay, a ganglion is where we have a whole bunch of these grouped together. So, okay, now neurons can have different structures depending on where they are and what they're doing. I just want you to be familiar with them in general. So they're multipolar neurons. So they have one axon and lots of different dendrites. These are the most common kind of neuron, these multipolar ones. So they're in your brain and your spinal cord. Okay, there are some that are considered bipolar. Okay, so bipolar, the axons or the cell bodies in the middle. Okay, and they're one axon, one dendrite. So they're found in your olfactory nerve, in your retina, in your ear. Okay, unipolar are going to leave. So they have a single axon coming away from the cell body. Okay, and there are some that are anaxonic, so they don't have an axon. They have lots of dendrites, and they're in the eye. Okay, I'm not too worried about you memorizing all of these. I just want you to know the basics. Like, they understand that there are different structures. Here again, if you look at, okay, now axonal transport. So what's going to happen is, because this is the cell body, this is where the endoplasmic reticulum and the ribosomes are. So this is where the proteins are going to be made. So the proteins have to be taken from the soma, so from the cell body, and they have to be transported down to the axon terminal or the synaptic knob. Okay, so axonal transport, so there's two-way passage. Okay, so there's anterograde, which means it's going to go down the axon away from the cell body, and retrograde means it's going back the other way. So just two different ways things can move. So anterograde, it's going forward. Retrograde, it's going back. Go back to this brain matter or... Okay, now, the structure of a nerve and a ganglion. Okay, so I want you to be able to... trying to find my notes. I want you to know the layers of this. So you should know the layers. So if you look at this picture, okay, so here's the epi. Okay, so that's like, if you think about the epi is always on the outside. Okay, then perineurium, okay, is going around here. So it's wrapping up a bundle. Okay, and the bundle is called a fascicle. Okay, and then the endo wraps a single axon or a single nerve cell. Okay, now a ganglion, and you can see ganglions here. Okay, a ganglion is a bundle, someplace where we have a whole bunch of cell bodies of an axon. So it ends up looking like a knot, looks like a bump. These are spinal cord matter. Ganglia look gray. Okay, Okay, so they're going to be gray. Have a better one. No. So the epineurium is going to be the outermost layer. Okay, then the perineurium, okay, is going to wrap up these fascicles that are inside of there. Okay, then the endoneurium is going to wrap up inside of there. Okay, so epi, peri, and then endo. Okay, and let me go back. Okay, so a collection of cell bodies along the length of a nerve. When we do spinal cord and the brain, you'll see examples of ganglion. Okay, but it's where I have it written out. Okay, now the synapse is the end. Okay, so the synapse is at the end of the transmissive part. Okay, so the synapse, it just kind of, I don't know how to describe it. It kind of just looks like a little knob. And what's going to happen here, okay, is this space that's called the synaptic cleft. Okay, so there's space in between it. So the neurons don't touch each other. They come close to each other, but they don't touch. So what will happen, okay, is these are vesicles in here, and these vesicles contain neurotransmitters. So the idea is that these vesicles with the neurotransmitters migrate to the ends of the synaptic bulb, and they exocytose the neurotransmitters out. Then on the next neuron, okay, there will be receptors, okay, that bind the neurotransmitters. So that's how they're going to communicate. Okay, so synapse. So they refer to things as a presynaptic neuron if it's before the synapse, and a postsynaptic neuron if it's past the synapse. So like the synapse is right here. Okay, but the synapse is the space in between. So things have to cross that space. So I hope that makes sense. So the neurotransmitters are in vesicles, okay, and they get exocytosed out. They cross the synaptic cleft, and then they come over and they'll bind to receptors on the next neuron. Okay, so these are the neural glial cells of the central nervous system. So neural glial cells are support cells. So they, like I always think, so nerve glue. Okay, so nerve glue, they're helpers. They're not excitable. Okay, so they can't be excited like a neuron, but they can go through mitosis. Okay, now we're going to talk about, I have them listed out on the other on the next slide, okay? So here's an oligodendrocyte, okay, astrocyte, looks like a star. Microglia are these little tiny cells. Epidermal cells are like epithelium, right? They're going to line. Okay, so let's keep going and we can come back to this picture. Here, these oligodendrocytes, okay, so here's the cell body, and then they're going to extend and they're going to wrap around the axons, okay, and they have different extensions coming around. I mentioned before that the cell bodies are going to be gray, okay, so here's cell bodies and they're gray, and then the axon is covered with myelin, and that's what makes the white components of the brain. Okay, so here's the gray, and there's the white. Okay, so in your central nervous system, okay, we have 4 types of neuroglial cells. Okay, so this would be in the brain or the spinal cord. Okay, so we have oligodendrocytes, okay, and you can think like octopus with the O and the O. Okay, they are what's going to form the myelin sheath. Okay, so, okay, astrocytes, those are the stars, okay, those are the most abundant. So they do a lot because they're so abundant, right? So they nurture, support, protect, and guide. Okay, so they're part of the blood-brain barrier. They're going to regulate the fluid around the cells in the brain. They provide structural support, and They assist in they're like nurturing, so they assist in the development of neurons. I kind of think of them as like nanny cells. Okay, the ependymal cells are going to line the cavities of the ventricles, and they'll produce cerebral spinal fluid. Microglia are actually macrophages, so they'll go around and they'll phagocytize, okay, or break down the garbage that's in there. Think waste that needs to get destroyed. They'll gobble it up and break it down. So, okay, now in the peripheral nervous system, okay, the microglia cells are satellite cells and neurolemmocytes. Okay, so satellite cells are the ganglia of the peripheral nervous system. They're going to insulate the PNS, so they insulate the cell bodies and they regulate the chemical environment. Okay, and then there are neurolemmocytes, which used to be called Schwann cells, but they try to get away from using people's names, and they myelinate or provide the myelin for the peripheral nervous system. So that's what's going to make the peripheral nervous system look white. Okay, so now what's the myelin sheath? Okay, so the myelin sheath is an insulating layer that's wrapped around the nerve fiber. Okay, so it's 20% protein and 80% lipids. Okay, so you got to have fat in your diet in order to develop myelin. Okay, now if I pretend this is an axon. Okay, what happens is the myelin is wrapped around here. So you can almost think of it like if you took a pencil and you rolled up the tube of toothpaste around it. I can't draw it. Kind of like this. That's how the myelin is wrapped around the neuron. Okay, so if this is the axon, okay, we have places where we have the myelin, and then we have spaces in between those. And what those spaces are called is nodes of Ranvier, or neurofibril nodes if you want to get away from the person's name. Okay, so neurofibril nodes, or the nodes of Ranvier, those are the spaces between the myelin. Okay, now, so the initial segment, I don't know what I have written here. The initial segment is the area before the first Schwann cell. Okay, so it would be up here. Okay, so it would be there. This is where the signals begin. So I showed you that on the other axon or neuron. Okay, myelination begins at the 14th week of gestation, and then it keeps occurring during infancy. So it's really important to have fat. That's why you give babies full-fat milk, okay, because you need lipids in their diet. Or not babies, but toddlers. Okay, so here's a picture. Okay, so here's the axon of the neuron. Okay, and then here's the Schwann cell, or the neurolemmocyte wrapped around it. Okay, so all of this is the myelin, and it's a cell, so there's the cell, the nucleus. The plasma membrane of the axon is called the axolemma, and then the cytoplasm in there is called the axoplasm. Just making it more detailed. Okay, now if we compare the myelin sheath between the peripheral nervous system and the central nervous system, in the peripheral nervous system, it's created by the neurolemmocytes, and you can see how the cells are wrapped around the axon. Okay, and in the CNS, they're kind of connected. See how it's more like a network? So the oligodendrocytes maybe put out fingers like gloves, and then the fingers, it's really long fingers, right? And then they're wrapped around the axon. So neurolemmocytes, which were also called Schwann cells, make myelin sheath in the PNS. In the CNS, it's the oligodendrocytes. So two different types of cells, similar job. Okay, so there are nervous system disorders that affect the myelin sheath, and a big one is called multiple sclerosis, so MS. MS is autoimmune disease, and what happens is the myelin gets attacked in the central nervous system. So it impairs sensory and motor function. And there's a whole bunch of different types. That's just an example. Another one is I can't say it right, Guillain-Barré syndrome. So it's an inflammation of the neurons, causes loss of myelin on the peripheral nerves, and it can cause muscle weakness. It can cause, lots of times when people get it, they start out with an acute flu-like illness. Since Zika virus has been a big deal, they actually think Zika, there's a relationship between people that are infected with Zika, they're more likely to get this syndrome. So the virus could be causing the inflammation or causing some sort of autoimmune problem. Okay, now speed of nerve signal. So how fast the information travels down the neuron totally depends on how big the fiber is and whether or not you have myelin. Okay, so if you have a small neuron that's not myelinated, the information is going to go relatively slow. If you have a small myelinated, the information is going to go faster. But if you have a large and it's myelinated, it's going to go super fast. So 120 meters per second. That's like you're not even thinking about what's happening. Okay, now regeneration of peripheral nerve fibers. The PNS can regenerate if the soma is intact and there's some neurolemma. So if there's some of the membrane around it is still intact. Okay, so they know all the steps that we have so much to cover. We're not going to go over all those steps, but there's lots of research going on in this. So if it's not damaged too bad, some things can regenerate, but it's not 100% sure thing. Okay, in the CNS, okay, things don't regenerate. They haven't figured that out yet. Okay, so if there's a stroke or spinal cord damage, that's not going to regenerate. Your brain can compensate and fill in holes where stuff is missing, but you can't grow new neurons. Okay, now, it's important when you talk about how the neural impulse is conducted, that you understand electrical potentials and currents. So the nervous system, it's a series of cells, and one cell is talking to another cell, is talking to another cell. Okay, it's not one big wire. It's cells talking to each other. Okay, it's based on electrical potential and electrical current. And it's important to realize that living cells have a charge difference. Okay, so the inside and the outside have different charges. So the inside of the cell is negative 70 compared to the outside. Okay, so the extracellular, there's a difference. Okay, so because there's a difference, a charge difference, the cells are considered polarized.